to access. Okay, should I start again, Courtney? Okay, I'll just start from there. So Falk has really been one of the thought leaders in this community. Really, I would say, I don't want to say the grandfather, but one of the first kind of folks who have helped us think through how the immune system naturally modifies antibodies to leverage different immunological functions. From the beginning of thinking about how antibodies work together as swarms, all the way through to thinking about how subclass, isotype, and Fc glycosylation shape the cytotoxic functions of immunoglobulin molecules, both in the context of protection as well as in pathology. Falk's work spans various areas of human immunology, from infectious disease to autoimmunity to oncology, and he really helps us understand the basic principles and the rules by which antibodies are able to interact with the immune system to drive these, very different types of immunological activities. So with that, we asked Falk to talk about a very specific question today, which we are so thankful that you've agreed to do this, Falk, but really to focus in on the idea that antibodies might function not only differently across different individuals, but also in a different way across different organs where they are fighting particular diseases, whether they are infectious, oncological, or autoimmune. And so with that, I'm going to pass the microphone over to Falk, who will take us on this journey, thinking about this unique question that we have not yet addressed in this series of talks. Thank you, Falk, for joining us today.
Well, thanks a lot, Galit, and thanks a lot to the organizers for this really kind introduction and allowing me to talk actually about one of our favorite topics, as I just mentioned, as Galit just mentioned, is really how cytotoxic antibodies work across organs. And I think there's really a kind of shift in paradigm going on, really how we think about these antibodies work. And this, of course, will be very important for designing also future approaches of how we design cytotoxic antibodies, of how we make them better, and what kind of effector cells are important and pathways are important to make them work in different organ environments. So just as a very, very brief introduction, to highlight again that cytotoxic antibody activity, while being kind of the standard thing that every one of us thinks about when you have a classic cytotoxic antibody in your mind, such as rituximab or Herceptin, which are used for cancer therapy, but actually more and more also for the treatment of a variety of autoimmune diseases. So here, of course, the organ environment becomes very important. If you think about lupus, if you think about arthritis and how these antibodies have to work in these organ environments, but actually also in cancer therapy, beyond the classic cytotoxic targeting of the cancer cell, but actually many of the immunomodulatory antibodies, just think about CTLA-4 antibody, have been shown, at least in certain tumor environments, to mediate their activity, at least in part via cytotoxic killing of regulatory T cells that are present within the tumor. And then, of course, we can also think about virus-infected cells, think about HIV, any kind of virus that hides in certain cellular reservoirs, where we want to target this kind of hiding virus reservoir via cytotoxic antibodies that can detect even few amounts of viral proteins expressed on the cell surface and then kill or address the virus repertoire. But again, these antibodies also may cause harm and damage. Think about SARS-CoV-2, where these antibodies then maybe target infected cells in the lung. And again, we need to understand how these antibodies then mediate cellular depletion, so cytotoxicity in these kind of organ environments. Now, in principle, and again, that is something that you find in textbooks. If you think about how can cytotoxic antibodies work, if you just take the word cytotoxicity, I mean, what happens? You inject an antibody and the target cell disappears, right? So this would be kind of the groundwork, the phenotype that we have. But the question, of course, as well, what kind of pathways are involved? And of course, there are antibodies that do that without any kind of effector function. So antibodies that have don't require the Fc portion, which I'm not going to talk about today. I'm going to talk about really cytotoxic activity in vivo that requires the Fc portion of the antibody, which is the dominant pathway, of course, in vivo. And here, basically, of course, the complement pathway may play a role, killing cells via membrane attack complexes, or classical ADCC working via NK cells and perforin granzyme mediated kind of killing. So this is kind of the groundwork, the pathways that you'll find in textbooks. And I'm not going to talk about the complement pathway today, but really want to focus on the ADCC, so the cellular pathways that underlie cytotoxic antibody activity. And really of course, you can do that with many types of different antibodies. So I'm going to focus on one specific antibody specificity, because this really is the hallmark or the kind of antibody that everyone relates to when it comes to textbooks and kind of model systems. And this is antibodies targeting B cells. And of course, what you can see is when you think about a patient, I don't know if you can see my mouse here, the cursor. But in case you can see it, so what is shown here in A, I can also use let me just use the pointer here. So what you can see here in A is that, what you can study in humans, and this is what you're limited to, is you can inject the antibody into the peripheral blood. You'll see the B cells will be opsonized, and then the B cells will be gone, right? So this is what you see. This is what you can study. It's much more difficult if you think about Herceptin, other kind of antibodies targeting solid tumors. You cannot look at it. You cannot follow it. So in the peripheral blood, this is what you can follow. And again, trying to recapitulate what's going on, then of course you can say, well, so I just take the peripheral blood, I put it in a test tube, I do exactly the same thing, I put in my cytotoxic antibody, and you'll see the same thing, right? The B cells will disappear. And the idea, of course, then was, and then you can, do the same thing, recapitulate it with isolated NK cells, you can recapitulate the same thing with isolated complement, and that, of course, has led to the assumption that a cytotoxic antibody activity, if you inject it against a certain target, it happens in that kind of environment. So in this case, in the peripheral blood, via the effector cells that are in there, and of course, the most effective effector cells in the peripheral blood that can mediate an ADCC reaction would be NK cells. Now, and that would lead also to a kind of model where we have kind of a seek and destroy thing, right? So you inject, if you take this model one step further and you think about a tumor, for example, or organ-resident autoreactive cells, single disseminated tumor cells, the idea would be, so we just have to inject the antibody, and IgG antibody gets into the tissue, it opsonizes its target cell, and then we would have kind of active hunting mechanism going on. So the NK cell or myeloid cell would migrate through the body and search for opsonized cells and then kill them, right? So that would make it very easy, actually, because then you only have to think about using the right antibody, the right isotype that gets to the right, that will find your cell. You won't have to take care of it. You could inject it, if you wish, directly into the tumor cell, but then everything else would be done by cells looking actively for opsonized targets. So this kind of seek and destroy mechanism. Now, however, what this model does not take into account is that, yes, we inject things in the peripheral blood, and we may opsonize, we may have our target cell in the peripheral blood, but the peripheral blood, of course, also flows through organs. And in organs, the whole situation becomes more complicated, because in organs then, additional cells, organ-resident cells, may have access to the opsonized cells that flow through the blood, right? And this could be tissue-resident macrophages, for example, in the lung, in the liver. Think about the bone marrow. So I'm going to talk about a couple of these organs. So we may have actually additional cells that can contribute to this kind of ADCC reaction. I'm always going to call it ADCC because what happens in the end is cells will deplete something, right? The mechanism of how this happens is nothing I'm going to relate to right now. So, but cells will disappear. So our initial question for actually a couple of years now is to really trying to figure out to what extent in vivo kind of opsonized cells residing in the blood or in a different organ where they become opsonized are really depleted in that organ environment, or if that happens completely somewhere else. Now, in order to answer that question experimentally in vivo, this is not straightforward, right? This is something that is kind of complicated because you need to find strategies that allow you to find, and this is what I put here on the strategy B, ideally what everyone would do these days, so think about, and we as well did, about pre-deleter strains that allow a cell type specific deletion of, say, activating Fc-gamma receptors. Now, the issue with that is that you can do that for NK cells, you can do that for a monocyte, but then again, you know, if you think about one step further, this will also deplete, for example, if you think about myeloid cells, this will deplete on the tissue-resident macrophages as well as on the monocytes in the blood. So teasing apart that kind of contribution of tissue-resident cells versus the cells that are coming from the bone marrow is not straightforward, and actually difficult to achieve with standard pre-deleted strains. So we decided to take one step back and actually look at a different kind of model system that is more crude, but allows you to answer a question maybe more easily. And this is using bone marrow chimeric mice. So because with bone marrow chimeras, right, you irradiate mice, you can delete the bone marrow, right? But you can spare the tissues. That would be our idea. And then if you use an approach, as is shown here, where you reconstitute mice that either have or not have Fc gamma receptors, you can come to a situation where you can get rid of Fc receptors on all the cells that come from the bone marrow. So your blood will be devoid of cells expressing Fc gamma receptors, but organ resident cells will still express Fc gamma receptors. So this allows you this initial kind of, distinction between where is an ADCC actually happening. And just to show you that this really works, this is shown here. So we use different kinds of titrations, we titrated irradiation, different irradiation dose. So we use 6 gray, 7 gray, 8 gray, and 9 gray irradiated mice, reconstituted them with bone marrow from mice where we can distinguish between CD45.1 and CD45.2. And what is shown here then is that you see it is regardless if you use 6 gray or 9 gray, you will always exchange all the cells that come from the bone marrow. However, when you look into the tissues, and I'm just showing you the liver here, but it's true for liver, lung, and skin, you can see that at the 6 gray radiation dose, you actually maintain all the organ resident cells while you completely exchange all the cells coming from the bone marrow. So this allows you exactly that kind of distinction where organ resident cells, for example, will still express Fc gamma receptors, and all of the cells in the blood, NK cells, myeloid cells, neutrophils, they lack Fc gamma receptor expression. All right, so what happens then if we do this exactly with this model system where Fc gamma receptor, where we use mice either sufficient or deficient for the common Fc-receptor gamma chain, and the common Fc-receptor gamma chain is the signaling molecule required for all activating Fc-receptors in mice. So by doing that, you can generate a situation where you knock out the function of all activating Fc gamma receptors. And what is shown here under B is that you can see actually we can generate exactly this situation where either all the bone marrow recipient cells lack Fc gamma receptor expression. So this is just shown here for the Ly6 high and low monocytes, also true for the neutrophils and NK cells. Whereas all the organ resident cells, just shown here as Kupffer cells, do express Fc gamma receptors. So we can create exactly this black and white scenario, Fc gamma receptors expressed on blood cells or organ resident cells. And then we just injected into these mice a CD20 antibody and looked at, when does it work? When do we see a depletion of B cells in the peripheral blood? And so what is shown here under D then is actually kind of the really surprising or really clear finding, nothing to be discussed, is that really only when you have Fc gamma receptors expressed on tissue resident macrophages, B cells are depleted. Whereas in the opposite situation, where all the effector cells, potential effector cells in the blood express Fc gamma receptors, nothing is happening, right? The opsonized B cells is floating by next to NK cells, myeloid cells, neutrophils. They just don't do anything, right? They don't bind to it. They may bind to it, but they don't kill it. So the killing happens via an organ resident cell. This is what this actually tells you. Now, of course, one could immediately argue, well, if you read a little bit about organ resident macrophages, the thing is that in mice, we know now clearly these are fetal derived, right? They are not continuously replenished by bone marrow. So this could be a very artificial situation because in humans, when we're young, we're kind of hunting aliens or kind of other soldiers. But when we're old, we may have still weapons, but we're carving pumpkins, right? This is what these two hunters here do, if you look on the right side. So you may still be in place, but you're no longer active. And what's also known that during inflammatory responses, and this is of course what we're exposed to once we're an adult, have been exposed to many inflammatory stimuli, these cells that sit there in the organs, which are fetal derived, may be exchanged by bone marrow derived cells, and they may do a completely different job. So what we actually describe here may be relevant for humans being very, very young, but not for adults, where this kind of exchange is going on. So we try to also account for this situation and use the following approach. So we again do this kind of bone marrow chimeric approach, where we can create this on and off situation. ADCC is on when all the organ resident cells express Fc gamma receptors. But what we can do now is we can, kind of mimic this stimulus where we have inflammation. So we just inject these mice with clodronate liposomes, which deletes all the phagocytes coming from the bone marrow and the tissue resident cells. Now this will open up the niche in the tissues and will allow the migration of cells coming from the bone marrow into these niches. Because we empty the niches, these cells are no longer there, so they will be filled with, ideally with the bone marrow derived cells, which will then turn this, ideally, that was our idea, this on situation into an ADCC off situation, because suddenly other cells are sitting in these niches that in this case no longer express Fc gamma receptors. So this would be kind of expected. But this situation here is the more interesting one, the off situation, where all the bone marrow resident cells here did express Fc gamma receptors, tissue resident not, because now these cells should migrate into the tissue. And if they settle down in these niches and have the same function, they should be able now to turn on this ADCC reaction. And another nice kind of side effect of this kind of approach is if you inject clodronate liposomes intravenously, it will not target lung macrophages, right? So the lung, the Fc gamma receptor expression on the alveolar macrophages, interstitial macrophages will remain the same. We will just exchange it on the liver. So let's see what happens. First, this shows you that this actually works. So this is kind of here staining of CD45.1 and 0.2. Again, where we show before treating with clodronate liposomes, we have all the kind of purplish cells in here. All the macrophages express CD45.2, which is coming from cells, for example, here not expressing Fc gamma receptors. Whereas when we do the clodronate treatment, wait for 4 to 6 weeks, we can see that all these niches now have been filled by CD45.1 positive cells, now expressing Fc gamma receptors. So this is the quantification here, but what is happening to an ADCC reaction? And well, actually, exactly that, what I just suggested to happen is we have now this kind of off situation before. This is what I showed you before. So no ADCC, despite all the monocytes and NK cells in the blood express Fc gamma receptors. Now we empty the niche, allow these myeloid cells to migrate into the niche where the Kupffer cells were, and suddenly we generate the on situation. Same mouse, right? Just clodronate treated, waiting until the bone marrow, until the niche is filled, and we can see ADCC is turned on, whereas in the opposite situation, ADCC is turned off. So indeed, this kind of organ, it is the organ resident cells that are important here for this activity. Now that we have actually an idea that this actually may happen in the liver, not in the lung, because what I just told you before, now we actually had the chance to go for a pre-deleter mouse, because there's a very efficient mouse that we got from Martin Julien, who created a CLAC4F3 mouse, which deletes selectively on Kupffer cells in the liver. Now, what we did is we now used an Fc-receptor 1 gamma chain flox mouse, crossed it to this mouse. And what you can see here is that this very specifically on Kupffer cells, as the wild type situation, down here deletes all the activating Fc gamma receptors. Inhibitory Fc gamma receptor remains the same. So organ resident macrophages in the liver, the Kupffer cells lack Fc gamma receptors, where all the other hepatic myeloid cells that are in there have a normal Fc gamma receptor expression pattern. So creating exactly the same situation, but very, very specific for the liver. So what is happening to ADCC? Well, the same thing, ADCC is gone. So B cells are no longer depleted if we specifically delete activating Fc gamma receptors on Kupffer cells. So telling us really that this is happening in an organ, and just as another control, we did a standard model that we also have platelet depletion, which is expected to be or thought to be happening in the liver. And again, you can see that the platelet depletion with an autoantibody is also abrogated if we specifically delete activating Fc gamma receptors on Kupffer cells. So what that tells us is that actually the B cells, and this also is true for B cells being opsonized in the spleen or in the peripheral blood, then despite the effector cells are present, they are not depleted here, but they have to migrate into the organ environment of the liver, where Kupffer cells, but not necessarily Kupffer cells, any cell type that sits in this niche, can have access to the opsonized B cells and kill it. And this could either be phagocytosis, trogocytosis, cell fragmentation, trophobtosis, whatever the mechanism will be, shown by all these other studies here. So I think this is one nice example of how this can happen. So what we actually would have rather, if this is true, we would have more a waiting for the prey model, really, not an active hunting model, but waiting. The macrophage sits there, it just waits for things to float by. If they don't float by, they will not get depleted despite they're opsonized. But if they come in close proximity, then actually your macrophage will deplete the opsonized target cell. So this is a very important concept now, and a really shift in the concept, because a macrophage does not migrate. It just sits there, it samples. So like this guy here fishing, you have kind of the fishing rod, but this has a certain kind of length. And if your fish is, beyond that and it doesn't move close to it, you will not be able to get it. So I think that's a very important take-home message. But now let's look into another organ, and this is the bone marrow, because the bone marrow really is an organ where it's highly complex. This is where many tumor cells have been shown to hide, where you get relapses. So how is an ADCC reaction happening in the bone marrow? Again, we'll stay with the B cells. I'm not going to go beyond the B cells today. So the bone marrow, this is just an exemplary slide here showing you that we have many different types of niches. Of course, we have the areas where the B cells recirculate through the bone marrow, where we have B cell development and different types of niches. So at least 2 or 3 different types of niches in this organ that are important that may determine how a cytotoxic antibody works in this kind of environment. All right, so just to show you a little bit, just trying to get rid of this is working. Let's see if this just need to get rid of, oops. Okay. See if I can start. Yeah, so I can start. So this is showing you a staining of this is a light shield microscopy staining of the bone marrow, staining B cells with an anti-CD20 antibody, because this is our target antibody, but with a mutated Fc, so it can no longer deplete basically B cells, and CD31. So what this shows you, this is kind of here the osteal bone. This is the bone. This is the blood vessels that go into the bone. So red stainings would be the B cells, and you can see that the B cells are sitting either in these blood vessels, the CD20 positive ones, or right next to this outer area here where we have the cortical bone in the bone marrow, where you would expect the mature B cells to sit. Again, very nicely shown down here. This is where our target cells sit in this kind of bone marrow niche. Now, in order to look at also a little bit at the timing, because we now talk about a kind of an organ that may not be directly or clearly accessible. All the cells may not be accessible for your cytotoxic antibody. So we injected this antibody, CD20 antibody labeled, into mice, C3 Fc-receptor gamma chain knockout mice, where we get no depletion, but we can label the B cells. And we can see that expectedly we label immature B cells with this antibody and the recirculating mature B cells in a very narrow time frame. So between 2 and 4 hours later on, this antibody staining is gone. If you look 1 day later, antibody staining is gone. Despite, again, remember, there is no depletion. So this may be just a normal turnover of the B cells that we see. But this will be our target cells that I'm going to refer to for the rest of the talk, the immature B cells and the recirculating B cells, which are the CD20 expressing cells. So let's look, and now, of course, I just told you that basically the liver can be a place where B cells can be depleted. Now, of course, we could have the effect here that immature B cells, which can also migrate, as you can see here, into the blood, and also the mature B cells that recirculate, of course, can have access to the blood. So if we see the depletion here, it could just happen also in the liver, and actually not in the bone marrow. Now, to make the distinction, we again now use this Kupffer mouse where we delete all the activating Fc gamma receptors on the Kupffer cells. But you can see, if you look at the bone marrow B cells, mature B cells, immature B cells, the B cell depletion works. So this is actually not happening via migration into the blood and then depletion in the liver. This is happening in the bone marrow. That's kind of an important control here, that this is not happening indirectly again in the liver. So this happens. B cell depletion of mature recirculating B cells, immature B cells happens within the bone marrow. So then, of course, to get an idea what are kind of the effector cells here, because we are now talking about an entirely new organ, we looked at different kind of Fc gamma receptor deficient mouse strains to see which kind of Fc receptors are important for the cytotoxic activity. And as we have seen for other studies, an individual activating Fc gamma receptor 1 or 3 or 4 they're not important, right? And an individual receptor is not enough. If you knock out one, ADCC still happens. What we can tell, although already, is that NK cells are not essential for this effect because NK cells only express Fc-gamma receptor 3, right? This would be the only cell type where we can say now, okay, it's not the NK cells that kill the B cells in the bone marrow. It's going to be other cells. To get to that in more detail, we now use double knockout mice, Fc-gamma receptor 1 and 3 double knockout mice, but you can see here B cell depletion still works of the immature B cells, a little bit less, but still works. Mature B cells still works. Only if we block Fc-gamma receptor 4, then the B cell depletion is really impaired. So we require, I would say, most of the activating Fc-gamma receptors, at least Fc-gamma receptor 4 alone, can do the job. We always do the reverse experiment where we knock out Fc-gamma receptor 1 and 4. And as you can see here, actually, Fc-gamma receptor 1 and 4 seem to be more important compared to Fc-gamma receptor 1 and 3. But again, R4 alone cannot do the job. So Fc-gamma receptor 1 and 4 seem to be the most important Fc-receptor for mediating this depletion of mature and immature B cells. So this would tell us this could be macrophages or non-classical monocytes, because these are the cell types co-expressing Fc-gamma receptor 1 and 4. Now, in order to see whether this effect, again, is mediated by phagocytic cells, we injected clodronate liposomes into these animals. And what you can see here also that it's quite clear, yes, so it must be phagocytic cells because clodronate is able to completely inhibit this B cell depletion in the bone marrow of both mature and immature B cells. So it is phagocytic cells. Now you might say, well, I know very well from experiments, if you do it in peripheral blood, you inject clodronate liposomes, it will deplete the monocytes and the macrophages, and the monocytes and organ-resident macrophages very efficiently. Now, interestingly, if you look in the bone marrow, and this is something that we have seen back in 2019, where we did a similar experiment, if you look in the bone marrow, clodronate does not deplete bone marrow classical monocytes or non-classical monocytes. It seems to quite specifically act on macrophages. Whether this is kind of because of access to clodronate liposomes, we don't know. But so we can actually rule out the monocytes as effector cells because this really acts on macrophages. And more specifically, if you look on subsets of macrophages, we can see that, and this is all F480 positive cells, the CD62L-GR1 positive F480 cells are also not depleted, but it's rather the CD62L-GR1 high, CD62L intermediate and low cells that seem to be depleted by clodronate liposomes. So this kind of study we did for a different kind of study where we looked at NK cell depletion, but that tells us already that it probably these kind of myeloid cells are very important. So we went on from this study and repeated this kind of experiment, injected clodronate liposomes, and again showing that yes, indeed, also a couple of years later, we see the same result. So this is the GR1 staining and the CD62L staining, this kind of red circle population, and these are all F480 positive cells. These are the ones that are depleted by clodronate liposomes. Furthermore, these are the ones that are phagocytic. So when we inject fluorescent beads, this is the cell population that can phagocytose beads, and this is the cell population that really will eat up fluorescently labeled liposomes. So also here, this high population, but it's really the largest population is this CD62L low, GR1 low, which is really phagocytic. Now, of course, that gave us a hint that indeed this could be a cell subset involved actually in this depletion of B cells in the bone marrow. Now, in order to look at that in more detail, we set up a more sophisticated staining panel, in this case using 23 different types of antibodies to further subdivide this Ly6T low, Ly6G low cell population down here. So this is the one labeled in orange and blue and red. And we can distinguish them by expression of TIM4. This would be the orange population here in CD11B. Now, all these three subsets, unfortunately, can be depleted very well by clodronate liposomes. So this kind of clodronate liposome staining, clodronate liposome experiment cannot tell us which ones of these is more important for this. They're all phagocytic, they can all be depleted, so they can all be involved in this ADCC reaction in the bone marrow. And most of them actually express Fc-gamma receptors 1 and 4, which are the critical receptors, right? I would say the least likely population from this experiment would be the red population here, because the Fc-gamma receptor 4 expression is really, really low on this subset, but they all express the high affinity receptor. Now, in order to get one step further, we also used now again this kind of irradiation model, because this allowed us to distinguish between irradiation sensitive, kind of tissue resident, bone marrow derived myeloid cells in there, and the ones that are more fetal derived or irradiation resistant, I would say. So again, this is a mixture. They're all F480 cells. It becomes really difficult to distinguish F480 positive cells now coming from the bone marrow and coming from fetal derived origins. But as I'm going to show you now, I think this kind of very low dose irradiation works again very nicely, because when we look at the three different substances, the orange one here, the TIM4 positive one, when we do this kind of crosswise chimeras with 6-gray radiation, we can see that this orange population seems to be at least 50% of the cells seem to be radiation resistant. So we cannot, using this low dose radiation, we cannot fully exchange them. Whereas the blue population here, CD11B high, and the red population can be fully exchanged. So this is what you would expect from a hematopoietic stem cell derived kind of cell that develops into a macrophage in the bone marrow. We can fully exchange them in both situations. So these cell types now, if we do again this Fc-receptor positive, negative kind of crisscross exchange, should be fully exchanged with cells here expressing Fc-gamma receptors or not expressing Fc-gamma receptors. But the orange ones should be not. Actually, they're very resistant to radiation. Now let's look what happens when we do this kind of crosswise bone marrow transplants. This is a situation where we use mice that have Fc-gamma receptors, where we put in bone marrow with where no Fc-gamma receptors are expressed. So in this situation, we can see that the mature B cells and the immature B cells are fully depleted. So this would mean, coming back to this kind of scheme here, that here all the blue cells, so the CD11B high and the CD11B low, TIM4 low, they've become fully exchanged to 90–95% by cells no longer expressing Fc-gamma receptors. So the only cells that still express Fc-gamma receptors are the orange ones here. So coming from this kind of scenario, a model system, this would indicate that indeed it is, at least to a large proportion, this TIM4 positive cells, irradiation resistant, that sit in the right place where they can deplete the mature B cells. The other way around, again, now all these blue cells, they express Fc-gamma receptors in the red cells, only the orange ones largely do not. We see a major impairment of depletion of mature B cells. For the immature B cells, a little bit more complicated, because here still we have some ongoing depletion also in this scenario, which could mean that if you look here where the immature B cells sit and the mature B cells sit, that some of these kind of TIM4 positive cells are actually sitting still close by the immature B cells here, and they can still deplete them despite the majority of them has also been exchanged for Fc-gamma receptor expression. So at least what that tells us, allows us to exclude these two cell populations, and makes it very likely that this very specific focus areas of ADCC are present in the bone marrow. Now, in order to stain for that, or kind of make that visible, we used a kind of Kawamoto tape method to cut the bone marrow without fixing, having to decalcify it. And as you can see, this method works very nicely. So this is the cortical bone, this is the whole bone marrow. And these mice have been injected with the dye-eye liposomes, so with the fluorescently labeled liposomes. And you can see this in these select spots of high phagocytic activity appearing here. So this tells us that it's really certain hotspots where highly phagocytic cells sit in the bone marrow environment that may then be the cells that deplete B cells. Now, of course, this is nice showing in a kind of static way, but we also wanted to see this in a highly dynamic way. So we went one step further and generated mice where we can actually look at the bone. We can actually follow in live how B cells become depleted. And this we did together with collaborators in Berlin, Anja Hauser, and they do this kind of operation. They put kind of a window on top of the bone where you can put in your dual photon microscope, and you can, and you put this here on top of the bone. You can look, you can insert then here your microscope, and you can look into the bone marrow. The mice are then healthy, the mice walk around, and you can look in the bone marrow and look in real time how B cell depletion is happening in the bone marrow. And so this is shown here. So the red cells now that are in here are TD tomato labeled, RFP labeled B cells, and the blue cells are CD169 positive macrophages. And as you may remember, probably not, so CD169 was one of the markers that is highly expressed on this TIM4 positive macrophage subset. So this would be the steady state situation. Let me see if I can actually get rid of my laser pointer again. And because this is also a movie, it's kind of hard to. Here we go. So let's start this again. So this is. Turn off.
Do you want to just go ahead, Falk? I don't know if it's
Yes.
stuck. Yeah.
So here now I can click on the move. So what you see here now is, so this was the situation before. This is the situation after we inject the antibody. And you can nicely see that basically what is happening here is the B cells in red are largely gone. You can see all these kind of orange kind of spots here, which is basically the situation where we only have debris of B cells going on. You can quantify that after different types of time points, and you can see very nicely here that the B cells have become disintegrated, are just little kind of particles sitting around which have been eaten up by the macrophages. So basically, as a summary, what that shows you is that now we've looked at two different types of organs, that what is happening if cells are opsonized in the blood, for example, the depletion of these cells, B cells, will happen in organs such as mediated by Kupffer cells in the liver. If you look at the bone marrow, we have a highly complex situation where certain subsets of macrophages, despite there's many macrophages, only certain subsets that mediate this activity. I think we could narrow it down to this kind of CD169 high, TIM4 positive subsets. But it really reiterates that it's kind of only very specific phagocytic cells that can mediate this activity in certain organ environments. And there has been a very nice paper. So this again reinforces that also in the bone marrow, we have this kind of waiting for the prey model. There is the macrophage sitting there. There's many macrophages, but only certain macrophages are really sitting in close proximity. And only these macrophages will be able to phagocytose an opsonized cell. And this has been shown in a paper by Philippe Bousseau in 2021, I think, where they used labeled tumor cells, and they showed that only if these tumor cells were sitting really in close proximity to a macrophage, they could be phagocytosed, again using a CD20 antibody, whereas if they're further away, they cannot be phagocytosed. So this explains very easily. And this also shows here that macrophages, he looked at macrophages at different time points after injection of these antibodies, they're sessile, they don't move, they just sit there, they can spread out their arms, again, like the fishing, like if you're fishing, you can, put out your, throw out the rope here, but that's it. That you have a certain distance where you can catch a target cell. If the target cell is opsonized further away, nothing will happen to the cell. So this explains very nicely niches that can occur in different organ environments where ADCC is just not happening, where tumor cells can hide. And my final slide that reinforces this is one of the reasons why we now go into different organs and kind of look at Fc-receptor and different organ resident cells, resident immune cells. And again, you can see incredible differences in the amount of Fc-receptors expressing throughout these different organs. For example, showing that in the skin, we have huge amounts of inhibitory receptor expressed on organ resident skin immune cells, such as Langerhans cells, dermal macrophages, or dermal myeloid cells. So compared to other organ environments, such as the lung or as the spleen, where we have a low level of this expression. Reversely, we have a super high expression of activating Fc-receptor 4, in this case, on kidney resident macrophages, whereas we have rather low level of other activating Fc gamma receptors expressed here. So this, and Fc gamma receptor 4 is not expressed at all during the steady state on dermal resident immune cells, right? So we have now really complex scenarios where not only the macrophages are not migrating, but they have different types of steady state Fc gamma receptors that contribute to this kind of ADCC activity. So reinforcing that, really understanding the organ environment is very important if you want to understand and make therapeutic antibodies better and make them work in different kind of niches. So that's it. Sorry, I went a little bit over time. Acknowledgement here to Pascal, who really did all the work on the kind of B-cell depletion model. We got the Fc-receptor flox mice from Jeff Ravage. Martin Julien provided us with the CLEC4F3 mice. All of this, apart from the last study that I showed you, is unpublished, is in the process of being submitted, so please don't distribute it too much, I guess. And funding was provided by the German Research Foundation and by an NIH grant together with Galit. It was a great pleasure to really look at these kind of organ-specific environments. So thanks a lot for your attention. I'd be happy to take any questions.
Thank you, Falk. That was wonderful. So I'm going to clap for everyone. And if there's any questions from the audience, please put them in the Q&A, or we can even maybe promote you to the panel. But that was really so elegant, Falk, as always. Really getting to the point, really showing us, what's happening deep in those tissues. So maybe I could start off with a couple of questions until folks start to post their own questions in the chat window. But I guess the first question I have is, so mouse versus humans, right? NK cells do have different properties, especially with their natural killer cells. And so I'm blown away at the fact that, NK cells are completely not critical for these types of depletion functions. Do you think that that's a species issue? Do you think that in humans there could be a role for NK cells, or are you convinced by the data that it's really a tissue resident macrophage depleting activity that dominates how we respond to rituximab?
So I think, what we're looking at here, so first of all, I think I would fully agree that a major difference between mouse and human NK cells is that, first of all, mouse, they express only one activating Fc-receptor. It's always Fc gamma receptor 3, that is clear. NK cells, I think, express lower levels. We also quantified NK cell expression, CD16 expression on mouse NK cells, and this is clearly lower. But I think this really relates to this kind of steady state situation. Because in the steady state, I mean, imagine how many NK cells do we have? So at the reactions that we look at here, this is a very fast, immediate reaction. And we have just a very few percentage of NK cells, right, in mice and in humans. And we have this is happening immediately. And this immediate reaction, I think, is happening, I think, in humans as well. I mean, Jeff showed it with he used the CD16 transgenic mice where we have human Fc gamma receptors expressed on mouse cells. And he also saw that it was not the NK cells now in the setting of a tumor. But I think the NK cells are very potent. We just need to get them to the right place, right? And this may just take a little longer.
In the human liver,
yes,
40% of lymphocytes are NK cells.
Yes, but they are floating through the blood, right? And they probably don't.
They're tissue resident.
Tissue resident in the liver.
So maybe that's a difference. I don't know. I don't know enough about mouse livers. So this is why, maybe
if you don't perfuse, you have quite a number. Definitely not that amount. So I would agree then, of course, if the human liver has that high amount of NK cells, surely they could contribute. Absolutely. I mean, the question is, would they have the access, right? Would they be in the right place? Of course, that is difficult to do, that kind of experiment. In the mouse, you can basically take out the liver, and this was done in 2005, and ADCC is blocked. I think what we will need in this kind of respect, and also with respect to the human liver, I think some kind of spatial transcriptomics, kind of high definition, looking at the niches are NK cells, if they are that present in the liver, in the human liver, do they have access to B cells? Are they in close proximity? Because I think don't think, I mean, I've seen stains of human livers and I've seen Kupffer cells. Again, I didn't have, you know, and many of the Kupffer cells are not actually in close proximity because there's only certain amounts of cells that are close to the blood vessels and then have access to the B cells that flow by, because the opsonized B cells don't migrate into the liver and then look for their effector cell. So I think understanding the niche, so I would fully agree. So understanding the niche in humans as well as in mice, with kind of more sophisticated, high resolution, spatial transcriptomics, whatever you name the method to really look at the niche will be very important. Though I think in other organs, such as, you know, in tumors, yes, there's tumors where we have lots of NK cells. Many tumors also know from humans we have always the myeloid cells. I didn't talk about tumor results today, but I wouldn't exclude the NK cells. But what I would think really is that, you know, approaches that people use, for example, attracting NK cells by putting IL-15 kind of cytokines on their antibodies that will still label their effector cell going into the tissue. I think this would be a highly important and highly efficient, because NK cells are super efficient in killing, right? That's absolutely clear. And the myeloid cells, what it tells us is that you have lots of niches where now cells can hide. Either we get the myeloid cells to migrate, and I think that's the macrophages to migrate. I don't think it's going to happen. So the much more important mechanism to improve on ADCC is getting the NK cells to where they're supposed to go. Yeah. And I think what the recent studies tell us on using CAR T cells, because they migrate, right? They go into the tissues, they're there, and they will find I mean, they've been highly efficient now in curing lupus. Amazing. So getting cells that actually can migrate where they're supposed to go, I think this is the much more promising approach, instead of now taking this data and telling us, well, we kind of have to stretch the myeloid cells to engulf the tumor cells, because either the other way around. So I'm fully with you. We need to understand more with respect to the species differences, but also we need to get the NK cells to where they're supposed to go.
Do you have a question, Joe? Because if not, I have another question.
Yeah, probably too simple, but I'll ask it anyway. In terms of, so this is amazing, and I want to really thank you, because I truly understood. I think I understood a lot of it. So, and this is pretty intense, and it's not my space. But so thank you. I'm wondering, this is all. I look at the B cell model that you're using as you said, it's kind of a tumor cell like model, and I'm wondering, how does this compare to, you know, tissue specific infections, and can you relate it to COVID, right, and lung and things like that? So that's all.
Yeah, no, absolutely. No, it's a very good question. So so we're using this kind of B cell model, and actually we're not using tumor cells here, injecting tumor cells. That's why I showed the last part here, where they used actually also the naturally occurring tumor cells from a multi-transgenic model. The problem is when you inject the tumor cells that you grow in vitro, they kind of downregulate all these complement regulatory proteins. So they suddenly become very much prone to being just depleted by complement. So that's why we're kind of staying with a natural model to understand the niches, but I fully agree. And I think this comes back to the situation, which I didn't show today, in the tumor cells, because in the tumor environment and also in the infection environment, you have inflammation, you have recruitment of many bone marrow derived cells to the site of infection, tumor growth, where now other subsets come into play. And this actually is the case. And suddenly this creates, but this is the second step now, right? Where we now can distinguish, for example, with inflammation, and we're trying to address this angle from the tumor cell perspective. If we have all these immigrating TAMs and NK cells and neutrophils, now to what extent do they play a role in combination with these tissue resident cells that are there and self-repopulating? And we could show back in 2017 that, for example, if a tumor grows in the skin, you really require the bone marrow derived cells migrating into the tumor to kill the tumor. Whereas in the lung, the alveolar macrophage myeloid cells in the lung are so active because of this kind of inflammatory context that they are mediating cytotoxic antibody activity against tumor cells, and it's not the tissue resident cells. Now, again, you know, other cells that become, with respect to infection, I think it becomes super interesting if you think about kind of infection with parasites that have more this kind of IL-4 prone kind of M2. I'm not supposed to say that word, but kind of M2-like whatever. Macrophage, people will kill me if ever, so don't spread that. I never said M2, but this kind of, skewing where different kinds of Fc-receptors are expressed, if then kind of other immune cells come into play. So all of that is an absolutely open question. It becomes more and more difficult to address. That's why we're also using humanized mice, where we have the human NK cells, normal expression level, these kind of things. So very good question. Everything open and many, especially with respect to pathology also, if you think about SARS-CoV-2, a very important question, not only with respect to clearing virus, but also with respect to pathology. Yeah.
And so maybe I can ask the question because it follows perfectly along with that from the audience. And so Falk, there was a question here that said, obviously, like nice talk, but the question really was about the tumor itself, and just give you a chance to talk about it because M2s were invoked in this question. And the idea really is the reason why you have poor depletion or monoclonal activity because of some sort of disruption of the macrophage populations in the tumor. So even when you recruit, is there some sort of localized inflammatory defect? And within a tumor, is there, a certain exhaustion associated even just intrinsically in the cells that reside there, when they come in, even without the M2 effects?
Yeah. So I think there's two answers to this question. If you think about a cytotoxic antibody targeting tumor cells itself, we and many others, it's mostly hidden in the papers. You can treat very early if you wait until the tumor has a certain size. The antibody still gets in the tumor, the macrophages are there, and then the cytotoxic antibody doesn't work anymore. At least it's no longer able to kind of significantly shrink tumor mass. So does that tell us, so there could be two answers to this question. As you said, the myeloid cells may be kind of exhausted and no longer be able to do it. I think what is a little bit arguing against that scenario is that if you take the immunomodulatory antibodies, such as CTLA-4, which deplete regulatory T cells in these tumors, they can do that even during late tumor growth. So and this is also mediated via myeloid cells, right? The cytotoxic activity against anti-CTLA-4 antibodies killing Tregs is myeloid cells, TUMs in the tumor. So that would tell you that actually the cytotoxic activity in principle is there, I think. But again, the cytotoxic T cells reside in different areas, and where much of the myeloid cells go, if the tumors are larger, you have kind of necrotic areas, you have hypoxia, this upregulates inhibitory receptors on macrophages in certain tumor areas. So I think the changing tumor areas, location, hypoxic areas, that allows a cytotoxic attack against the tumor is no longer active. It could very well be that in these areas, the myeloid cells, because of hypoxia and again, changing their phenotype may no longer be as phagocytic, but there is other areas, and I think that's good news, where myeloid cells, where the regulatory T cells reside, and they can kill them. So it's not the entire kind of myeloid cell subsets are inactive in the tumor, but some may be. Again, understanding areas within the tumor. Very complicated, but very good question.
Last question, Falk. Thank you very much. That was wonderful. So last and quick question. So you're showing a vanilla IgG here, right? So I'm assuming it's not really modified. Do you think that the biology then changes? Is it a different effector cell if you now, modify the glycan or add point mutations, or are the rules always conserved and it is the local effectors that do most of the hard work?
Yeah, so I think here, of course, so we used an IgG2A antibody, non-modified, but we used an IgG2A because this is really the only, and here I would fully, be along your lines, this would be the only correlate to the human IgG1 or IgG3. Yes, it would be. So kind of if you use and we use that because this has really the broadest kind of interaction with all effector cells as human antibodies. If you go to a mouse IgG1, this suddenly binds super high to the inhibitory Fc-receptor. So there the situation becomes even more prone to kind of myeloid cells and inactivating them. I think if you go to these other mouse subclasses, you would be more prone to getting into something mouse specific. That's why we stayed with the IgG2A, which is really the most active. But yes, so you could make this more active by also, taking away fucosylation, which would trigger, again, in the mouse, this would be probably a little bit of difficulty comparing that to the human, because this will trigger an even higher affinity for Fc gamma receptor 4, which is not expressed on mouse NK cells. So that's why we stayed also away from that, because the plain IgG2A has high binding to NK cells, myeloid cells. So this is closest to the human situation. But again, in the human situation, taking an fucosylated one, which works better, this would attract more NK cell activation also. So this would be also arguing you would get, in the human situation, more NK cell attracted or activity to the tumor cell.
Wonderful. Okay, thank you, Falk. So I think we're 1 minute away from the end of the seminar. So you just absolutely perfect presentation, wonderful question. It was great to have you join us and to end this seminar series on such a high note. So with that, there is a link to the webinars on the chat window. So for all those in the audience, and hopefully this has been a fun experience for everyone on the calls. Falk, I hope we'll see you at the meeting to start to summarize these activities. And I want to thank the co-chairs, Lisa and Joe, and of course, Trey and Courtney for all their help in organizing this meeting. And hopefully this will be the beginning of a wonderful community focused on Fc effector function. Lisa and Joe, if you want to say anything finally, but thank you all.
Thanks so much. Perfect.
Thank you. Bye, guys.
Take care. Bye-bye.
Thank you.
Thanks.